study broadly, and this is what I'm going to touch on. So in terms of this talk, I'll give you a little bit of background about the phenotype data I'm collecting, a summary of what we've done so far in terms of NeuralNet between Cambridge and Colchester, and then dig a little bit deeper into that HPO data, as in the phenotype data, and talk about the overview with East of England, and think a little bit more about how the study broadly fits into the future. So the human phenotype ontology is the HPO that I'll be referring to throughout the talk. It's a standardized vocabulary that's been developed by the Robinson Group in 2008 for primarily rare disease researchers, because it's based on OMIM entries. Currently supports terminologies for over 7,000 diseases, and as you'll see in the next slide, each term is a relationship. The roots are the grandparent terms, as in the very broad terms, and the leaves are the children terms. So if you see that you have a patient with bilateral sensory neural hearing impairment, that's your most specific term in this is a relationship. And it goes back through the ontology up to phenotypic abnormality, which is the broadest term. And what we prefer is having as specific a term as possible in rare disease research, because that enables better gene-disease relationships to be constructed. Why that's important is because researchers like myself think about phenotype similarity scoring and how we can apply it for studies where we're trying to diagnose undiagnosed rare disease patients. So this illustrates how similarity scoring works. Essentially, you have a query patient, say you have if you look down at C, and this is an image lifted from Kohler et al.'s paper from 2009. So you have your query patient who's got these two terms, so downward slanting palpebral fissures and hypertelorism, and you've got two reference data sets for two different syndromes, and you've got Noonan syndrome and Oppitz syndrome. And essentially, Noonan syndrome fits best with your query patient, giving a higher similarity score because the terms are most specific, and it's based on information content, so as in how specific that term is in your reference data set. So coming back to NeuralNet, we've got, so this data that I'm talking about is looking at the Cambridge and Colchester data correct up to December 2023. So we might have a couple more patients by now. So we have 41 patients up to December 2023, and 39 had complete sets of records, including the Qualtrics data. We've broadly got 5 diagnoses, and the HPO data has been gathered both from Qualtrics, the questionnaire that's been filled out, and the electronic healthcare records on both sides. So Cambridge records have been explored by a bioinformatician, so the EPIC records, and then the Colchester Evolve records have been searched by myself. And when you look at the HPO terms and just look at the number of HPO, the number of unique terms that we've collected for each patient, as you can see, the Colchester data from the electronic records, we have a lot more, and that's because I've basically just gone through each of the scanned Evolve records. And the Qualtrics HPO counts are fairly similar, but perhaps a few more in Colchester. And this is digging a little bit more into the HPO terms. So sorry if the writing's a little bit small on the y-axis, but essentially the graph to your left is the Qualtrics data, and the graph to the right is the clinical data. And as you can see, there's a lot more sort of CP specific terms in the top 20 terms collected in the Qualtrics data for both Cambridge and Colchester. and sort of premature birth and respiratory failure, intellectual disability, for example. In the clinical data, as in those collected from the electronic health records, there's a little bit more in terms of specific terms such as tiptoe gait, delayed speech and language development, frequent falls, for example. In terms of Colchester and Cambridge, so 1 second. Sorry if you can hear in the background my daughter screaming. It's just quite late in Sri Lanka. Sorry about that. So in the data by source, Colchester is on the left-hand side and Cambridge is on the right-hand side. And I think the one thing I might direct your attention to is that our top sort of collected term is intellectual disability in Colchester versus spasticity in Cambridge. And this is looking at all that data, so Qualtrics and the electronic health records. We do have in Cambridge intellectual disability featuring, but that's a little bit lower down on our top 20 terms list. So that makes a difference in terms of performing that phenotype similarity scoring within cohorts. So this is where I've essentially lumped all that data together for the Cambridge and Colchester patients, performed a similarity score where I've looked at phenotypes sort of between Colchester and Cambridge to see whether there's any clustering, and given the study IDs at the sort of x-axis, and you can see that the orange cluster, which is the Cambridge cluster, seems to cluster quite well together, and that's probably because they have quite a narrow phenotype, and then the Colchester patients seem to be a little bit more spread out, and the Cambridge patients don't seem to cluster that well. So when there's these big blue patches, that's essentially saying that there's quite a low phenotype similarity score between patients. And the cohorts therefore don't seem to cluster that well. But I haven't applied any statistics here because we have quite a small sample set. But visually you can see that there are some clusters and there are clear differences between the cohorts. When you look at the Colchester data and break it down by, because I suppose something we're quite interested in is the demographics of the population we're serving. The patients who've been recruited are mainly in more deprived, mainly from more deprived areas, and this is looking using their postcodes. So you have 4 out of the 21 patients recruited through Colchester who are actually in more of the sort of least deprived areas. Out of these 21 patients, we have 1 patient with a diagnosis. We have 16 out of the 21 who have a label of intellectual disability or global developmental delay or sort of autism or ADHD spectrums. The rest, so the remaining 5 have isolated gross motor delays. Well, and I should, explain that the graph here shows the count of HPO codes by the index of multiple deprivation, where 1 is the most deprived area and 10 is the least. Maybe there might be a little bit of a correlation, but again, it's too few data points for us to perform any statistics on this. So this then brings me on to East of England data, because we're thinking about demographics and whether we're representative of the populations we're serving. And this is data shared by Dr. Gemma Thiloka, who represents the East GLH. So interestingly, since whole genome sequencing testing was made available at Cambridge GLH, 2,150 rare disease tests have been carried out till May 2023. There were 299 unique clinicians requesting these tests, of whom 89 were pediatricians, and within that there are 10 who are community pediatricians. So this to point out that this is data from across the board from adults and pediatrics. Also useful to note that out of the 19 trusts requesting whole genome sequencing, SNEFT is in that top 5. So we're doing quite well in recognizing the need. Given that we're now performing studies where we're thinking about genomic testing more and more, it's important for us to keep up to date with what's going on in our field. So we've undertaken a scoping review, at least the start of a scoping review, to look at genomic testing in community pediatrics. And unfortunately, we can't find any that are relevant to the practice in the UK. There are some studies where they look at genomic testing for developmental delay, especially in the USA. But there's one study ongoing till 2025, which is evaluating the NHS Genomic Medicine Service for pediatric rare disease. But we won't have those results for some time. So should we be thinking about reviewing our current genetic testing practices in community pediatrics, looking at uptake, the types of indications and so on? And if we are, we should be also thinking about what we're doing with that data. So this is a little schematic I've drawn because I suppose it's helpful to know where we are at. And I think where we are at, from my viewpoint, is that we are generating a number of variants of uncertain significance, so in orange, because often we have limited data for the rare variants that we generate, or limited clinical phenotypes for the patients we are sequencing. And that leads to us having this uncertainty whether that variant is causing disease or not. So this leads us to needing more data. And that data is from electronic health records or radiology or further family history, or variant data, so pathogenicity evidence, whether that's functional studies or sort of matchmaking type studies with other international colleagues. It might also be a case of time, because children who are being sequenced may not have those phenotypes in place for us to have that full picture. The same goes for if we have a diagnosis, and here you have in the blue corner, you have a variant of interest with the matching phenotype at that time, which confirms the diagnosis. However, we might not be able to prognosticate the child or give further information to the family, just because we don't know which route that genetic diagnosis is going to take, whether they're going to have a very severe outcome or a milder spectrum of that disease. And again, we need to think carefully about how we automate the process of gathering that additional clinical data. I think I'm leaving that as an open question because it's something I'm still looking at, and there are no easy answers at this point. And with that, I'll end. So I wanted to thank you for listening. Thanks to all the patients and their families for taking part, and my collaborators and mentors. Just listed some of the people who are involved in this work, and my mentors at the bottom who've helped me in understanding phenotype similarity scoring as well. Thank you.